Today I will be telling you about the steps of the lytic cycle and the lysogenic cycle, and I will start by talking about the lysogenic cycle. The first step of the lysogenic cycle is when the phage binds to the surface of host cell and ejects its genetic material. The second step of the lysogenic cycle is when phage DNA is incorporated by crossing over into a specific site on the host cell's chromosome. The third step of the lysogenic cycle is host cells copy their DNA and divide many times, creating a large population of cells with phage DNA, quote unquote, hiding in their genome. The fourth and final step of the lysogenic cycle is environmental signals can trigger the phage DNA to leave the host genome and initiate the lytic cycle. And now I will be transitioning to the lytic cycle. The first step of the lytic cycle would be the attachment, in which the T4 phage uses its tail fibers to bind to specific receptor sites on the outer surface of an E. coli cell. The second step of the lytic cycle is the entry of phage DNA and degradation of host DNA. And in this, the sheath of the tail contracts, injecting the phage DNA into the cell and leaving an empty capsid outside. The cell's DNA is hydrolyzed. The third step of the lytic cycle would be the synthesis of viral genomes and proteins. And in this, the phage DNA directs proteins and copies of the phage genome by host enzymes using components within the cell. The fourth step of the lytic cycle would be the assembly. And in the assembly, 3 separate sets of proteins self-assemble to form phage heads, tails, and tail fibers. The fifth and final step of the lytic cycle would be the release. And in the release, the phage directs production of an enzyme that damages the bacterial cell wall, allowing fluid to enter. And there, I just completed the explanation of the lytic and lysogenic cycle.